Today, we are undertaking, well, a monumental task.
We really are.
We're plunging past the surface of the human brain and descending into the foundational architecture of the structure that controls, plans, and processes everything we experience, the cerebral hemispheres.
It's an essential journey. Our mission today is surgical, it's detailed, and it's highly technical, but we're going to present it in a way that gives you a genuine shortcut to neuroanatomical literacy.
Right.
We're going beyond just the simple mapping of function and diving into the literal hardware. I mean, how are the operating systems, the neurons, organized into layers?
And what does that structure tell us? What are the implications of that organization?
Exactly. What does it tell us about the input and output requirements of a specific brain region? And perhaps most fascinatingly, how do all these complex, specialized regions communicate via the intricate, massive highway system of white matter?
This deep dive is all about the blueprint. We are treating the brain today like, I guess, the most complex piece of computing architecture ever designed.
It certainly is.
When we discuss the cerebral hemispheres, we're talking about the largest component of the human brain, which is functionally designed to do, well, three main things. First, take in information from the external world.
What anatomists sometimes stiffly call teleception.
Right, a bit formal for us. We'll just call it sensory input.
Yeah.
Sight, sound, touch.
Good idea.
Second, we have the cortical centers for actually processing all that input. And third, the colossal white matter system for connectivity, which is what allows for global computation.
And we absolutely have to remember the internal environment. These hemispheres, they aren't just shells of cortex.
No, not at all.
Internally, they cradle the deep gray matter structures. We're talking about the basal ganglia, responsible for motor control and habit formation, the diencephalon, which includes the crucial thalamus and hypothalamus.
A great relay station.
The great relay station, exactly. And surrounding all of that, you have the convoluted, wrinkled sheet of the cerebral cortex itself.
So, functionally, we can look at these hemispheres as operating on two major tiers, right?
Exactly. Tier 1 involves the specific cortical centers, those primary zones, defined by their direct input or output roles.
Okay, so give me an example.
Think the primary sensory areas V1 for vision, A1 for audition, and the primary motor area M1. They are handling the raw data flow, the direct inputs and outputs.
And Tier 2?
Tier 2 involves the high-order association regions. These are the areas that integrate all that raw data. They're linking sensory input to memory, to emotional history, to planning, to executive control.
This is where the magic happens, so to speak.
This is where truly complex behavior, abstract thought, and emotional regulation reside. And understanding the architectural differences between these two tiers, the sensor-motor units and the association units, that is the key to understanding complex human perception.
Okay, let's unpack this enormous structure. We're starting on the outside, looking at the surfaces. Let's talk about those iconic wrinkles.
Let's do it.
When you look at the hemispheric exterior, the first thing that strikes you is the folds. The cortex isn't smooth at all.
No, it's formed by these deeply folded extensions of gray matter. And these are not random wrinkles. They are the result of intense evolutionary pressure to maximize surface area.
And the vocabulary we need here is essential. The valleys are the sulci.
Or fissures, if they're particularly deep.
Right, and the elevated ridges are the gyri. So why is this folding so important? Why not just have a bigger, smooth brain?
Well, it's a pure space maximization hack. If the human brain were smooth, it would require a head about the size of a watermelon to house the estimated 16 to 20 billion neurons in the cortex.
That's hard to imagine.
It is. So by folding the gray matter, the cellular bodies, we significantly increase the surface area available for cortical computation without requiring an impossibly large skull. In fact, only about a third of the cerebral cortex is actually visible on the surface.
The rest is hidden away.
The rest is hidden deep within the sulci.
What's truly remarkable is that this isn't a static structure, but one that develops dynamically over time. We can actually track when the major features appear.
We can, and it's one of the hallmarks of prenatal and early postnatal development. The first, most prominent sulci, the landmarks we rely on, like the central sulcus and the deep lateral fissure.
The Sylvian fissure.
The Sylvian fissure, exactly. They start appearing relatively early, around the 28th week of gestation. This is the scaffolding being laid down.
But the finer texture, the smaller details, continue to be refined after birth, right?
Precisely. The secondary sulci, those smaller, more individual wrinkles, they continue to form postnatally, generally between the 7th and 13th months.
So it's still developing well into the first year of life.
And beyond. Even more complex structures, particularly the gyri of the temporal lobe, are known to continue their development and folding, albeit at a much reduced pace, potentially until a person is in their late teens.
Wow.
This just highlights that the brain's physical structure, especially the cortical surface, is an ongoing developmental project. It's sensitive to both genetic and environmental influences well beyond infancy.
That timescale is remarkable. From the second trimester well into adulthood. Let's use those folds to orient ourselves. Let's start with the external or lateral surface, which is the view most people picture when they think of the brain.
The lateral view. Okay, it's defined by the 4 major lobes and the 2 main separating fissures.
So the first big one is the lateral fissure.
The lateral fissure is that massive, deep groove that separates the temporal lobe below from the frontal and parietal lobes above. It's essentially the horizon line on the map of the brain.
And the other major landmark.
Then you have the central sulcus, running roughly vertically. And this is the great dividing line between action and sensation.
So it separates the frontal lobe, which is all about motor control and planning?
Anteriorly, yes, from the parietal lobe, which handles sensation and spatial awareness, located posteriorly.
And within the frontal lobe, what specific landmarks do we see there?
There we see the superior, middle, and inferior frontal gyri, and they run horizontally. These house our complex cognitive and language centers, like the prefrontal cortex and Broca's area.
And we see a similar pattern in the temporal lobe.
We do. In the temporal lobe, you have the superior, middle, and inferior temporal gyri. And while the exact pattern of these smaller gyri can vary significantly from person to person, it's almost like a fingerprint, the locations of the main sulci are surprisingly consistent across all human brains.
Now let's rotate the brain and look at the medial surface. This is the interior wall where the 2 hemispheres touch. The architecture here is dominated by one massive white matter structure.
The medial surface is absolutely defined by the corpus callosum, that enormous C-shaped band of commissural white matter that connects the 2 halves.
The Great Bridge.
The Great Bridge. And wrapping around it, we find the cingulate sulcus and the cingulate gyrus. The cingulate gyrus is a primary component of the limbic system, linking emotion, motivation, and memory, and it forms this critical ring of cortex right on that medial wall.
And if we move further back on that surface?
Towards the posterior end, where the parietal and occipital lobes meet, we find the prominent parieto-occipital sulcus. This is a major boundary marker on the medial surface.
And what about near the top?
Just above the corpus callosum and near the central sulcus, we find the paracentral lobule. This is the critical medial extension of the primary motor and somatosensory cortices.
So this is where the representations for the legs and feet are located.
Exactly, which makes sense given their position at the top of the body map.
And then the complex underside, the inferior surface. This is the part that interacts directly with the bony structure of the cranial floor.
The inferior surface is a complex, almost triangular patchwork of cortical tissue. The frontal lobe rests entirely on the floor of the anterior cranial fossa.
And the back of the brain?
Moving posteriorly, the back portion of the brain, so the occipital lobe and parts of the temporal lobe, it rests on the tentorium cerebelli.
Which is that dural fold separating the cerebrum from the cerebellum.
Exactly. And this complex underside features the junction of the orbitofrontal cortex, the temporal, and the occipital lobes. This area is notorious for having sulci and gyri that are highly individualistic.
Why is that?
It likely reflects the high degree of specialized processing occurring there, particularly related to emotional context and complex object recognition.
We've covered the exterior. But the folding process doesn't just create mountains and valleys, it actually hides an entire island of cortex, the insula. This is where the structural complexity gets even more layered.
The insula, or the island of Rel, is a prime example of the brain reserving critical computational space. It is a roughly triangular piece of cortex situated deep within that lateral sulcus.
And it's completely concealed from the external view.
Completely. It's covered by the opercula. Those are the flaps of the frontal, parietal, and temporal lobes that cover it. To see the insula, you literally have to peel back those lobes.
Why hide it? What does that geographical isolation suggest about its function?
Well, its location, deep at the junction of 4 major lobes, perfectly reflects its function as an interoceptive and integration hub.
So it's a meeting point.
It is. It's separated from the surrounding cortical regions by the circular sulcus. Structurally, it's divided into a larger anterior part, which breaks down into short gyri, and a smaller posterior part with longer gyri.
And functionally, what's happening there?
Functionally, it is a nexus for gustation, which is taste, visceral sensation, your gut feelings, pain perception, and integrating emotional context, often linked to the amygdala with conscious sensation.
So hiding it ensures that it's constantly processing this primal integrated input.
Precisely. It makes it less susceptible to the noise of the primary sensory cortices, allowing it to generate what we consciously feel as gut reactions.
It's the brain's secret weapon for self-awareness and emotional processing. It's fascinating how the brain uses its internal geography, those folds, fissures, and hidden islands, to organize function.
It really is.
But to truly understand how it processes information, we need to go smaller, moving from gross anatomy down to the cellular level. This is where we uncover the universal blueprint.
This is where things get really interesting.
Okay, let's zoom in 1,000 times. We are now looking at the cellular fabric of the gray matter, the cerebral cortex. This is the hardware that makes computation possible. We are talking about millions upon millions of specialized cells.
Indeed, the cortical tissue is a highly structured matrix composed of neurons, the communicators, and glial cells, the support structure, alongside a dense network of blood vessels. But for now, we need to distinguish between the two primary functional classes of neurons.
The first major group are the principal neurons. These are the ones doing the broadcasting, sending signals long distances.
Correct. The principal neurons are the output cells. They use the major excitatory neurotransmitter, glutamate. The vast majority of these are the pyramidal cells.
And they get their name from their shape.
They do. They are instantly recognizable by their pyramidal or triangular shape. They possess a characteristic apical dendrite pointing toward the surface and basal dendrite spreading out laterally. They are the chief projection neurons.
So they're responsible for sending information out of the cortex.
Either to distant cortical regions or down to subcortical areas. They're the long-distance communicators.
And the second type of principal cell.
Those are the spiny stellate cells. They're typically smaller, more star-shaped, and they're covered in dendritic spines.
And their role is different from the pyramidal cells.
Very different. Unlike the pyramidal cells, which are primarily long-distance messengers, the spiny stellate cells are usually local circuit neurons. They contribute to the specific, localized processing loops within the cortex itself. They make up the dense computational fabric of the receiving layers.
So we have the broadcasters, pyramidal cells, and the local processors, spiny stellate cells. Both were excitatory, firing off signals using glutamate. Now, what about the second major group, the interneurons? They sound like the crucial modulators, the system regulators.
Precisely. Interneurons are local cells. Their axons rarely leave the cortical region where the cell body is located, and they are the inhibitory element in the network.
And they use a different neurotransmitter.
They use GABA, gamma-aminobutyric acid, as their neurotransmitter. Their function is absolutely fundamental. They ensure that the network doesn't run away with itself.
What happens without that GABAergic inhibition? What would that look like?
Well, you get synchronization failure and runaway excitation, which manifests clinically as seizures. The delicate balance between glutamatergic excitation from the principal cells and GABAergic inhibition from the interneurons is the very foundation of synchronized, coherent cortical function.
And these interneurons aren't all the same, are they?
No, they're incredibly diverse. We have smooth stellate cells, but also highly specialized inhibitory neurons like basket cells, which wrap their axons around the cell bodies of pyramidal cells to inhibit their firing.
Very direct form of control.
Extremely direct. And then you have chandelier cells, which specifically target the initial segments of pyramidal cell axons. They're essentially acting as ultra-precise circuit breakers. Their diversity just reflects the need for incredibly sophisticated local control.
That's a highly sophisticated traffic control system, managing the communication of billions of specialized cells. Now, let's move to how these cells are structured into a repeatable layered pattern.
This is the architectural marvel we call the six-layer laminar organization. It is often referred to as the universal rule of the isocortex, or neocortex, which makes up roughly 90% of the human cerebrum.
So every single piece of cortex that isn't part of that ancient memory structure follows this rigid blueprint.
That's the rule. This organization is defined by cytoarchitecture, the differences in cellular makeup and density across the depth of the cortex. The study of this architecture is called laminology.
And these 6 layers, labeled I through 6, they define the flow of information.
They do. They define where specific types of input land and where different types of output leave. And while the thickness of each layer varies depending on whether the region is sensory, motor, or association, that basic 6-stack structure is always there.
Let's start at the very top, the outermost layer, layer 1.
Layer 1, the molecular or plexiform layer, is the most superficial, lying just beneath the pile surface. It's unique because it's relatively acellular.
Meaning it has very few neuronal cell bodies.
Exactly. Its primary composition is fibers. The terminal axons ascending from deeper layers and, critically, the apical dendritic tufts, the highest tips of the large pyramidal cells from layers 3 and V. It also contains the horizontal cells of Cajal.
So layer 1 is primarily the zone where output cells from the deep layers send their dendrites to receive final modulatory inputs. It's a connection zone.
Precisely. It's a convergence zone for superficial connectivity. During early development, this layer is also famous for hosting the Cajal-Retzius cells.
And they have a really important job, right?
A crucial job. They secrete a protein called relin, which is essential for guiding neuronal migration to ensure all the deeper layers form correctly. Once their job is done, they largely disappear, leaving layer 1 as primarily a dendritic and axonal field.
Moving down, what defines layer 2?
Layer 2, the external granular layer, is densely packed. It's characterized by a large number of small, tightly packed neurons, mostly small, non-pyramidal granular cells, interspersed with smaller pyramidal cells.
And its function?
Functionally, layer 2 serves primarily as an association layer for local communication. It communicates heavily with layer 3 and adjacent cortical regions, processing and refining information locally before it's passed on to long-distance circuits. You can think of it as the local area network.
And layer 3 is where the conversation goes long distance.
Layer 3, the external pyramidal layer, is dominated by medium-sized pyramidal cells. And sometimes, due to its complexity, it's broken down into sublayers, 3 thea, lycae, I, as, ake. This is the major source of all cortical projection fibers.
So anytime two distinct regions of the cortex need to talk to each other.
The signal originates in layer 3. Whether it's across the Great Divide of the corpus callosum, using commissural fibers, or just across the hemisphere, using association fibers, it handles the long-distance feed-forward communication necessary for integrated thought. It's the essential networking layer.
Now we hit layer 4. If layer 3 is networking, layer 4 is the primary sensor input hub.
That is the perfect analogy. Layer 4, the internal granular layer, is the primary input layer of the entire cortex. It is dominated by those excitatory, spiny, stellate neurons we talked about.
And this is where information from the thalamus comes in.
This layer receives the vast majority of thalamic input, all the specific processed sensory information that has been filtered through the thalamus. Layer 4 acts as the initial integration point for external data.
So if I'm looking at something, that raw visual data from the lateral geniculate nucleus lands right here in layer 4.
It lands almost exclusively in layer 4 of the primary visual cortex, which is BA17. And this relationship gives us a rule. If a cortical area is heavily sensory, its layer 4 will be exceptionally thick and dense with these input-receiving neurons.
And the opposite would be true for a motor area.
Exactly. In a purely motor area like BA4, layer 4 is often quite thin because its job isn't to receive sensory data, but to generate commands.
And the counterpoint to the input layer is the command center, layer V.
Layer V, the internal pyramidal layer, is the primary output layer. This layer contains the largest pyramidal cells in the entire cortex, including the famous BET cells in the motor cortex. This is the ultimate output structure.
So it's sending commands out of the cortex to the subcortical world.
It is. Layer V has immense functional significance because it's the main source of the corticostriatal tracts to the basal ganglia, the corticobulbar tracts to the brainstem nuclei governing facial and head movements, and most critically,
The corticospinal tracts.
Yeah.
The ones that go all the way down the spinal cord.
the very ones. The commands to move your body. If you want to move a muscle, the signal originates right here in layer V. Naturally, this layer is the thickest and most prominent in the primary motor cortex, reflecting its role as the dominant effector pathway.
Finally, we reach the basement, the boundary layer, layer 6.
Layer 6, the multiform or polymorphous layer, is the deepest layer, and it just sort of blends into the white matter below. It contains a mix of cell shapes, hence the name polymorphous.
And what's its function? It's not just a border.
Not at all. Functionally, layer 6 plays a critical role in control by completing a feedback loop. It projects heavily and specifically back to the thalamus.
Wait, why project back to the thalamus? The structure that just sent the information up in the first place.
It's the cortex exercising regulatory control. By sending signals back to the thalamus, the cortex can modulate and regulate the flow of sensory information before it even reaches layer 4.
So it's like turning a volume knob on the sensory input.
That's a great way to put it. It allows the cortex to prioritize or suppress specific data streams, influencing attention and filtering based on the brain's current needs or state.
So the 6 layers represent a vertical flow chart. I for dendritic input integration, 2 and 3 for intercortical conversation, 5E for sensory intake, V for subcortical output, and C for feedback control. Understanding this blueprint is fundamental to everything else we'll discuss today.
It truly is the Rosetta Stone of cortical function.
We've built the house, the 6 layers. Now let's see what kind of traffic flows on the roads within it. The flow of information isn't arbitrary. It adheres to highly specialized rules based on the source and destination of the signal.
Absolutely. The input to the cortex, the efferent connections, can be segmented into 3 major classes, and each has a distinct pattern of termination across that laminar structure we just described. This specificity is crucial for computational efficiency.
Let's start with the non-thalamic inputs. These sound less like specific data streams and more like environmental regulators.
That is the essential distinction. The non-thalamic subcortical afferents originate from key nuclei in the brainstem and basal forebrain. These include the locus coeruleus,
For norapamine.
the Refi nuclei for serotonin, the substantia nigra and ventral tegmental area for dopamine, and the basal forebrain for acetylcholine. These inputs do not carry specific sensory data. They use powerful neuromodulators.
And you mentioned they terminate broadly, but especially in layers by 2 and 0, why they're...
Right. The reason for that broad distribution is that their function is to regulate the state of the cortex. They influence things like overall cortical excitability, alertness, attention, arousal, and mood.
So they're setting the tone for the whole system.
Exactly. By landing in layer I, they affect dendritic input, and by landing in layer 6-6, they affect thalamic feedback. They change the gain or the responsiveness of the entire circuit rather than delivering specific content. For instance, norepinephrine from the locus coeruleus helps focus attention by sharpening the contrast between important and unimportant signals.
Okay, now compare that to the thalamic afferents, the primary data feed.
These are the primary sensory and motor regulatory inputs. They carry specific, processed data visual, auditory, somatosensory. As we detailed, they are the main feed-forward input to the cortex.
And they terminate overwhelmingly in layer 4.
Overwhelmingly. That dense, specific termination ensures the initial raw processing of the data is localized and efficient.
And they also have that secondary link to layer 6, which feels like a very smart design choice.
It is brilliant because it allows for immediate regulatory control. The fibers from the thalamus that terminate in layer 4 are the driving input. But the secondary termination in layer 6 consists of fibers that establish the reciprocal connection, enabling the cortex to send signals right back to the thalamus to modulate future input.
So the cortex is an active participant in sensory processing, not just a passive receiver.
Precisely. It's constantly adjusting its own inputs.
Finally, we have the cortical afferents, the internal conversation between different cortical regions.
These connections, coming from layer 3 of another area, also follow specific laminar rules. The standard long-distance feed-forward connections usually terminate in layers 2 and 3.
So they're providing contextual associated information to the local processing network.
That's right. However, feedback projections signals returning to an earlier stage of processing. They tend to terminate in the superficial layer A and the deep layer V. It's thought to be a way of regulating the final output command based on higher-level context. That precise layered termination makes the laminar organization even more critical. The layer dictates the type of communication, whether it's raw data, long-distance context, or global state modulation.
Now let's quickly review the output. We know layer V is the commander, but let's review the full spectrum of efferent connections.
The outputs are beautifully segregated by destination. It's very organized. Layer 3 provides the output to other cortical areas, initiating those long-distance cortical conversations.
Then layer V.
Layer V is the major effector, projecting to all the subcortical motor structures, the basal ganglia, the brainstem, and the spinal cord. This is the physical command structure.
And layer 6 closes the loop.
Layer 6 provides the regulating output, projecting exclusively back to the thalamus, completing that corticothalamic feedback loop we just discussed.
And the chemical language of this entire operation remains fixed. Glutamate and GABA.
Correct. Glutamate is the major excitatory transmitter used by all the principal neurons, the pyramidal and spiny stellate cells. It drives rapid transmission and, crucially, is essential for synaptic plasticity.
which is the physical mechanism underlying learning and memory.
Exactly. Pyramidal cells are constantly releasing glutamate to signal downstream neurons.
And GABA, the essential brake pedal.
GABA is the main inhibitory neurotransmitter used by that highly diverse population of local circuit neurons, the interneurons. Its constant, precise inhibitory action ensures synchronous, efficient activity and prevents the runaway excitation that characterizes seizure activity. The complexity of cortical computation relies entirely on the subtle, moment-to-moment interplay between these two forces.
We've been discussing layers, which are horizontal divisions. But the most important organizational principle, the true functional unit of the cortex, is actually vertical. This is the concept of the cortical column or module.
This moves us from pure anatomy to true computational insight. Experimental studies, particularly in sensory cortices, demonstrated that the brain doesn't process information spread out across the horizontal layers like a liquid.
Which is how I used to picture it.
Right. Instead, it processes information focused like specialized computing racks. Neurons are organized vertically into functional columns or modules that span all 6 layers, from the surface right down to the white matter.
Can you give us a concrete example of this?
Yeah.
Because it's a bit of a mind-bending concept.
Of course. The classic examples come from the Nobel-winning work of Hubel and Weissel in the visual cortex, in V1. They discovered that if you stick an electrode into V1 and drop it straight down, perpendicular to the surface, all the neurons in that vertical path across layers 2 through VIO will share the same physiological property.
So they all respond to the same thing.
Exactly. For example, they might all respond only to a line segment tilted at exactly 45 degrees, or they might all respond only to light entering the left eye. These are the orientation columns and ocular dominance columns.
Wait, so within an area barely the width of a human hair, we're talking 150 to 300 microns, you have a fully dedicated processing unit for one specific feature.
That's the key insight. The columnar organization is the fundamental unit of cortical information processing. Raw input lands in layer 4. It gets processed and refined as it moves vertically through layers 2, 3, and 6, and the integrated focused output is generated by layer V, all within that tiny dedicated module.
That ensures maximum efficiency.
It does. It allows the brain to integrate specific features, like a single point of touch or a specific line orientation, before communicating the result to other modules. It's highly parallel processing.
This dramatically changes how we visualize the cortex. It's not just sheets of paper stacked on top of each other, the layers. It's a million tiny, highly focused computer chips running in parallel.
And this vertical organization is what makes the cortex so adaptable. The same 6-layered structure, the same columnar principle, can be applied to handle touch in the somatosensory columns, vision in the orientation columns, or audition in frequency columns. The hardware is universal. It's the information encoded within the module that's specialized.
The variation in the thickness to those 6 layers leads us directly to the famous maps. The brain isn't uniform. Regions are structurally and functionally specialized, and the original definitive mapping came from Corbinian-Brodmann over a century ago.
The Brodmann map, published around 1909, is just a foundational piece of work. It divided the human cortex into approximately 50 distinct areas, BA1, BA2, and so on. And what makes this work so enduring is that it was purely a work of anatomy.
He wasn't testing function in living subjects. He was staining dead brains and looking through a microscope.
Exactly. The division was based entirely on subtle differences in cytoarchitecture, the microscopic layout, the cell density, and the relative thickness of the 6 laminar layers. For instance, in one area, layer sole might be paper thin, while in an adjacent area, layer 4 might be massive. Brodmann categorized these changes.
And the incredible significance is that this purely anatomical observation became a functional roadmap.
The correlation is astonishing. Areas defined histologically by Brodmann later proved to be precise functional units. For instance, Brodmann Area 4, BA4, is the primary motor cortex.
Right.
Its cytoarchitecture is defined by a massive cell-dense layer V, housing those largest pyramidal cells, reflecting its pure output function, and a very thin layer 4.
Contrast that with a sensory area.
Contrast that with BA17, the primary visual cortex, which has an incredibly thick layer 4, reflecting its massive input needs from the thalamus, and a very thin layer V because its job is sensory input, not motor output.
This provides a reliable histological framework for understanding specialization across the entire surface, lateral, medial, and inferior. It's the original index for function.
It remains the gold standard, although modern techniques are revealing even finer subdivisions. The map includes key language areas like BA44 and 45, which is Broca's area, and the sensory areas like BA3, 1, and 2 for somatosensation. It just demonstrates that the thickness of the layers is adaptive to the region's primary role.
The six-layer structure, the isocortex, defines the cognitive bulk of the brain, but there are older, evolutionarily conserved parts that don't follow that rule. This distinction is crucial for our next sections on memory and emotion.
This is the narrative thread we need to pull on. The evolutionary history of the cerebrum is literally laid bare in its layered structure. The isocortex, neocortex, with its definitive, fully developed six-layered structure, makes up the vast majority of our cortex and handles our highest cognitive functions: language, abstract planning, and sensory discrimination.
But the parts of the brain related to more primal functions, smell, gut feelings, basic memory encoding, are ancient.
They are. And they're housed in the allocortex, or the old cortex, which has fewer than 6 layers, typically only 3. This ancient structure is found exclusively in the olfactory cortex and the crucial hippocampus.
So these systems are tied to primal survival mechanisms.
And fundamental memory processes, which reflects their older evolutionary origin.
But the modern cognitive brain, the isocortex, needs to talk to the ancient survival brain, the allocortex. How does that transition happen? Is there a bridge?
There is. It happens through the periallocortex and perisocortex. These are the transition zones. They exhibit intermediate structures, typically moving from 3 to 5 layers, that bridge the allocortex and the isocortex.
And where do we find these zones?
They're found in critical areas like the paraepicampal gyrus and the cingulate cortex. Functionally, they deal with complex transitional cognitive functions. For instance, taking a raw memory signal from the hippocampus and integrating it into a 6-layered association cortex for conscious recall and emotional context.
So they're the anatomical manifestation of the integration of primal survival with modern cognition.
That's a perfect way to describe it.
The olfactory system provides the most striking example of this older architecture, the paleocortex, because its pathway is fundamentally different from every other sense we possess.
The key structural difference is that olfaction bypasses the main sensory relay station. The pathway begins with the olfactory bulb and tract. The bulb's output, carried by mitral and tufted cells, projects directly via the olfactory tract to specific cortical areas.
Completely skipping the usual thalamic stopover.
Completely skipping it.
Why is that unique? I mean, every other sense, vision, audition, touch, is rigorously filtered and relayed by the thalamus before it hits the cortex.
It speaks to the evolutionary age of smell. Because olfaction is such a primal sense, often tied to immediate survival detecting poison, finding food, identifying mates, it needed a direct route.
A priority line.
A priority line, exactly. And its primary target is the paleocortex, specifically the piriform area, sometimes called BA51, located on the inferior surface.
And what defines the paleocortex?
It is defined by its 3-layered structure: a molecular layer, a diffuse pyramidal layer, and a multiform layer. This is the primary olfactory cortex.
And the signal doesn't stop there.
No, it also projects to the olfactory tubercle and the periamygdaloid cortex. This rapid, direct projection path ties smell immediately to the limbic structures, memory and emotion, which explains why a scent can trigger such a vivid, instantaneous emotional memory faster than almost any other sensory input.
So the architecture of the olfactory system explicitly links one of our most primal senses directly into our emotional and memory command centers, bypassing the higher-order filtering we apply to vision or sound. That's a perfect segue into the structures that define our emotional world.
Into the limbic system.
The limbic system is where that ancient 3-layered cortex is put to work, dictating our emotions, survival drives, and the mechanisms of memory.
The concept has a rich history. Initially, Broca defined the limbic lobe based on its anatomical ring-like structure around the brainstem, which she initially connected to olfaction.
Right, which is where the older term rhinencephalon or smell brain came from.
It is, but it was Papez who really gave it its modern functional identity.
Papez identified the core circuit that linked emotion and memory.
Yes, involving the cingulate gyrus, the hippocampus, and their connections through the anterior thalamus and the mammillary bodies. Today, we view the limbic system as a much more complex, distributed functional network. It's involved in emotional behavior, learning, memory consolidation, and autonomic control.
So we're talking about a functional network that integrates deep structures like the amygdala and hippocampal formation with cortical components like the cingulate cortex and their complex relays through the diencephalon and hypothalamus.
And critically, most of these cortical components, the cingulate, the parahippocampal gyrus, are either allocortex, 3 layers, or periarchocortex, that transition zone, which reinforces their evolutionary status as the machinery linking primal drives and environmental evaluation with conscious thought.
Let's start with the amygdala. It's often simplified as the fear center, but it's really the brain's ultimate threat detector and emotional evaluator.
The amygdala is a dense, almond-shaped complex of nuclei located deep within the rostral part of the temporal lobe, situated just superior and slightly anterior to the hippocampus. Its structure is defined by its 3 main groups of nuclei, which reflect a sophisticated input-processing-output flow.
Okay, so describe the input section. Where does the information come in?
That would be the large basolateral group. These nuclei receive incredibly rich, complex cortical input from nearly all sensory association cortices. The neurons here are large and loosely packed, acting as a highly sophisticated association region.
So it's an integration hub.
It is. When you see a shadow move quickly, the basolateral group integrates the raw visual input from the occipital lobe with contextual memory and spatial awareness from the parietal lobe to determine, is this a threat? This is where sensory input gains its emotional relevance.
So the basolateral group determines what emotion is appropriate. What about the medial group?
The medial group is smaller and maintains strong links to the primitive sensory systems, particularly the olfactory bulb, and has deep connections with the hypothalamus. This just reinforces that primal connection between smell and survival behaviors.
And the most important part from a behavioral standpoint, the output.
That is the central group. This is the command center for emotional expression. The central group projects massively to the brainstem nuclei and the hypothalamus.
And this system governs all the visceral and autonomic output related to emotion.
All of it. When the basolateral group determines a severe threat, the central group activates the fight-or-flight response, driving your increased heart rate, the hormonal stress response, freezing behavior, and immediate defensive actions.
It's the central switchboard for survival reflexes.
Yeah.
Given its role, its connectivity must be astonishingly broad.
It receives input from almost every sensory system, the sensory cortices, the thalamus, the olfactory bulb, and it sends projections to the prefrontal cortex, which is critical for the regulation of emotional expression.
So that's deciding whether to act on the central group's immediate command or override it with planned, sophisticated behavior.
Exactly. The amygdala is also crucial for fear conditioning learning to associate a neutral stimulus with a threat.
What happens when this crucial structure is damaged?
Damage, particularly bilateral damage, can lead to a severe disruption of emotional processing. A classic condition is Hirschsprung syndrome, where calcification or lesions impair the amygdala's function.
And what are the symptoms?
Patients exhibit severe difficulty in assessing and processing emotional stimuli, especially fear and threat. They might recognize a face but fail to identify the fear expressed on it, leading to dramatically blunted emotional responses and highly inappropriate social behavior because their internal threat detection system is malfunctioning.
If the amygdala is emotional evaluation, the hippocampal formation is the mechanism for laying down new conscious memories.
It is the absolute engine of declarative memory. It's located deep within the medial temporal lobe, forming this complex concave structure that spirals inward.
What are the 3 crucial elements we need to know here?
The structure consists of the dentate gyrus, DG, the hippocampus proper, which is divided into 4 subfields, CA1, CA2, CA3, and CA4, known as cornuamonis or Ammon's horn.
And the third part?
And the subiculum, which includes the precibiculum and parasubiculum. This serves as the primary output zone of the formation.
The crucial insight here is that memory encoding relies on a highly ordered, one-directional flow, the trisynaptic circuit. Tell us how that information travels.
The trisynaptic circuit is the physical pathway essential for long-term potentiation, LTP, the cellular mechanism of memory encoding.
Okay, so walk us through it. Step 1.
Step 1, the entry point. Sensory and contextual information, pre-processed by the cortex, first enters the hippocampal formation via the adjacent entorhinal cortex. This input projects to the dentate gyrus, DG, via the perforant pathway.
And what's the DG's job?
The DG is thought to perform pattern separation, taking similar inputs and making them distinct, which is crucial for memory specificity.
So that's the first synapse. Where does it go from there?
From there, the dentate gyrus sends its output to the hippocampus proper, CA3, via specialized, powerful axons known as mossy fibers.
And the second synapse is from CA3 to CA1.
Right. CA3 neurons then project to hippocampus proper, CA1, via the Schaffer collaterals. CA3 is heavily involved in pattern completion, retrieving a whole memory from just a fragment.
And the final step, the exit point.
Finally, CA1 projects to the subiculum, which acts as the major output relay, sending the processed and consolidated memory information back out to the entorhinal cortex and eventually to the neocortex for long-term storage.
That precise stepwise flow is what allows the brain to take a transient sensory event and convert it into a durable, retrievable memory trace.
Its functional role is strictly tied to declarative memory, the memory of facts and events, so episodic and semantic memory. This is famously illustrated by patient HM, whose bilateral removal of the medial temporal lobes, including the hippocampus, resulted in devastating deficits.
Which were?
The result is severe anterograde amnesia, the complete inability to form any new declarative memories, and significant retrograde amnesia, the loss of memories immediately preceding the damage. This proved the hippocampus is necessary for memory consolidation, though not for long-term storage itself.
And chemically, it's a glutamate powerhouse, right?
It is entirely dependent on glutamate to drive that synaptic plasticity. The perforant pathway, mossy fibers, and Schaffer collaterals are all glutamatergic, and the balance is maintained by localized GABAergic inhibition, ensuring that synapses are selectively strengthened or weakened during encoding without causing explosive activity.
We've established the six-layer rule, the fundamental unit, the column, and the architecture of the ancient brain, the allocortex. Now we can apply that framework to the four major lobes, seeing how the universal blueprint is specialized regionally.
Let's do it. We'll start with the biggest. The frontal lobe is the largest lobe in the human brain, and it really represents our highest evolutionary achievement.
It's a control center.
It is. It's responsible for executive functions, complex planning, skilled voluntary movement, and social regulation. It is essentially what gives us personality and foresight.
Let's start with the immediate action center, the motor strip.
That is the primary motor cortex, BA4, located in the precentral gyrus, just anterior to the central sulcus. Its sole job is to execute voluntary movements.
And it's organized somatotopically.
In the famous motor homunculus, a distorted map of the body, the face and mouth are represented laterally, and the legs and feet are represented medially, dipping onto the medial surface. This region has a ridiculously thick layer V, housing those massive BET cells, reflecting its powerful direct output capability.
Interior to that, we move into the planning areas. This is the difference between doing and deciding to do.
This is the premotor cortex, BA6, BA44, 45. It's involved in planning and sequencing movements, often based on external cues. BA6 is split into the supplementary motor area, SMA, and the pre-SMA.
And what do?
They're crucial for generating movements from memory or internal intent, independent of immediate sensory input. This is where you practice mentally before executing a complex action.
And this area also contains the critical engine for speech production.
Indeed. BA44 and BA45 together form Broca's area, the primary center for the motor execution and syntax construction required for language production. Damage here results in non-fluent aphasia, where speech is effortful and broken.
We also have the frontal eye field.
Right, BA8, which controls voluntary, goal-directed scanning movements of the eyes.
Finally, we reach the prefrontal cortex, the PFC, the pinnacle of human cognition.
The prefrontal cortex, PFC, covering areas like BA9 through BA12, is the ultimate executive center. It is defined functionally by its ability to hold and manipulate information over a short period, what we call working memory.
And it's also responsible for cognitive flexibility.
And its role in cognitive flexibility, complex planning, goal setting, and filtering distractions.
What's the implication of damage to this area? I think many people have heard of Phineas Gage.
Damage to the PFC can leave intelligence intact, but severely impairs judgment, social behavior, and emotional regulation, as tragically demonstrated by cases like Phineas Gage. The PFC integrates input from the limbic system for emotion and the sensory cortices for the environment to determine the most appropriate context-sensitive behavior. It's the highest expression of human self-control.
Okay, let's cross the central sulcus. We move from motor output to sensory input, entering the parietal lobe.
This is the primary receiving station for touch and body sense.
The primary somatosensory cortex.
BA3, 1, and 2, located in the postcentral gyrus. It receives dense, direct thalamic input, primarily to layer 4, for all sensations originating from the body. touch, pressure, pain, temperature, and proprioception.
Which is your sense of body position in space.
Exactly. And it is also organized somatotopically in the sensory homunculus, which mirrors the motor mac directly across the central sulcus.
So if you touch your finger, the sensory input lands exactly where the motor output from moving that finger originates, just across the street.
That spatial relationship is crucial for fast reflex arcs and coordinated action. Beyond that primary sensation, we also find the secondary somatosensory cortex, OP2, deep within the operculum, which contributes to higher-order processing and linking sensation to motor memory.
And the rest of the parietal lobe is dedicated to defining our spatial reality.
The extensive parietal association cortex is critically involved in spatial processing. It integrates visual information from the occipital lobe and somatic sensory information from the post-central gyrus to construct a coherent, integrated map of where our body is, where external objects are, and how we move through space.
And damage here can have really bizarre effects.
Damage to the right parietal lobe, for instance, often results in hemispatial neglect, where the patient completely ignores the entire left side of the world, demonstrating the parietal lobe's role in directing and maintaining attention across space.
Moving to the posterior pole, the occipital lobe is almost entirely dedicated to our highest bandwidth sense.
The entry point is the primary visual cortex, V1 or BA17, which is located deep within the posterior part of the brain, surrounding the medial calcarine sulcus. It receives direct, highly organized input from the lateral geniculate nucleus of the thalamus, landing in its massive layer 4.
This area is not just a receiving screen, it's highly organized, right?
Highly. It's organized retinotopically. A precise, point-for-point map of the visual field is laid out on the cortical surface. Importantly, this map is segregated. The upper visual field projects below the calcarine sulcus, and the lower visual field projects above it.
And the processing itself is vertical.
As we discussed, yes. View 1 is structured into functional columns that process input from one eye, ocular dominance, and specific angles, orientation columns.
Once V1 has done its initial processing, the information explodes into dozens of surrounding areas.
These are the extrastriate cortices, V2, V3, V4, V5, etc. These areas surround V1 and process specific visual features. For example, area V4 is heavily specialized for color and pattern recognition. Area V5, or MT, is specifically tuned for processing motion.
And this information then splits into two great streams.
It does. The dorsal stream, the Ware-Howe pathway, runs up to the parietal lobe for spatial awareness and action guidance. And the ventral stream, the What pathway, runs down to the temporal lobe for object and face recognition.
Finally, we turn to the temporal lobe, a massive hub for hearing, object recognition, and long-term memory integration.
The primary receiving station here is the primary auditory cortex, BA4142, located deep inside the superior temporal gyrus, within Heschel's gyrus. This is where sound first arrives from the medial geniculate nucleus.
And it's organized tonotopically.
It is. It contains a spatial map of sound frequencies.
Surrounding that primary area is where we unlock sound's meaning.
That's the temporal association cortex, which includes, adjacent to the parietal lobe, Wernicke's area. This region is critical for complex auditory processing and, most famously, for language comprehension.
And damage here results in Wernicke's aphasia.
Right, fluent but nonsensical speech. The ventral part of the temporal lobe is also the terminus of that visual what stream, making it essential for object and face recognition.
And we circle back to the insula, that hidden island we discussed earlier, now contextualized functionally.
The insula is truly one of the most fascinating hubs. It connects to the limbic system, the temporal lobe, and the frontal lobe. Because it processes gustation and visceral sensation interoception, it forms a central link between the body's internal state and conscious thought.
So when you feel anxiety in your stomach or recognize that a decision feels wrong,
the insula is highly active. It integrates emotional signals from the amygdala and physiological data from the brainstem, helping you make decisions based on what we call gut feeling. This makes it critical for emotional regulation, conscious awareness, and complex decision-making processes.
We've established the specialized processing units, the gray matter. Now we must address the infrastructure that makes global cognition possible. This is the white matter, the massive communication highway system.
Right. White matter is the system of myelinated axons, organized into large bundles or fasciculae or tracts that connect distant parts of the nervous system. The myelin sheath, that fatty insulation, is what gives it its white color and, crucially, dramatically increases the speed and efficiency of communication. It enables rapid, long-distance thought.
And we categorize these tracks into three types based on the connections they forge.
Projection, commissural, and association fibers.
Projection fibers are the vertical tracks, the longest cables, connecting the cortex down to the subcortical world, the brainstem, spinal cord, and thalamus.
The defining anatomical bottleneck for all projection fibers is the internal capsule. This is a massive, dense bundle of fibers located deep within the hemisphere, nestled between the thalamus and the basal ganglia. It's shaped like a V or a boomerang when you view it in a horizontal section.
And what's running through this tiny, concentrated space?
Everything critical. It carries nearly all the motor output from the cortex, the corticospinal and corticobulbar tracts, and all the primary sensory input to the cortex, the thalamic radiations. It also carries the auditory and optic radiations to their respective cortical areas.
So because all this absolutely critical information is squeezed into such a small area, damage here is catastrophic.
The internal capsule is clinically one of the most dangerous places to suffer damage. A small stroke here can result in devastating, immediate, and widespread sensory loss and paralysis on the opposite side of the body.
That concentration of function is why it's so vital to understand its specific parts.
We divide it into the anterior limb, the genu, which is the corner, the posterior limb, and the retro and sublenticular parts. For instance, the major motor tracts run predominantly through the posterior limb, while the auditory and optic radiations run through the sub- and retrolenticular parts.
Let's discuss another major projection bundle, one vital to the Papez memory circuit, the fornix.
The fornix is the main efferent or output bundle of the hippocampal formation. It is a striking C-shaped arch of fibers that wraps around the thalamus, arches backward, and then sweeps downward.
And what's its main job?
It serves as the primary connection route, linking the hippocampus for memory encoding back to the mammillary bodies of the hypothalamus, which are crucial for memory consolidation, and the septum. It ensures that new memory data can be broadcast and integrated into the broader limbic system.
Okay, now for commissural fibers. These are the horizontal links, allowing the two specialized halves of the brain to share information and synchronize activity.
The unparalleled structure here is the corpus callosum. Think of it as the world's most massive, high-speed fiber optic cable. It contains an astonishing 150 to 200 million highly myelinated axons. It is the defining feature of interhemispheric communication.
How is it structurally organized? It's not just one big bundle.
No, it's divided anatomically into 4 segments: the rostrum, the genu, the anterior bend, the vast trunk, the body, and the splenium, the bulbous posterior end.
And this spatial organization dictates connectivity.
It does. The anterior sections connect the frontal lobes for planning and executive function. The trunk connects the motor, parietal, and temporal lobes. The splenium connects the posterior, parietal, occipital, and temporal lobes, including the crucial visual and auditory association areas.
The synchronization is what allows us to experience the world as a unified whole, right?
Absolutely. The need for synchronization is why, in patients where the corpus callosum has been surgically severed, split-brain patients, you observe these striking disconnections where the left hand literally doesn't know what the right hand is doing because the sensory and motor information from the two hemispheres cannot be integrated.
And the second major commissural pathway.
That is the anterior commissure. It's a much smaller, curved tract located anterior to the fornix. It plays a specialized role connecting the olfactory bulb, the olfactory nuclei, and specific parts of the temporal lobes.
And it crucially links the two amygdalas.
Yes, linking the two amygdalas and the surrounding parietal cortex. This provides a direct, low-latency communication channel for emotional and primitive sensory context between the hemispheres.
Finally, we move to the internal connectivity, the association fibers, which ensure communication within the same hemisphere, connecting specialized modules across great distances or just adjacent to each other. These are crucial for language and abstract thought integration.
These tracks are essential. They're what take information processed in V1 for vision and send it forward to the frontal lobe for planning, or link auditory processing in Wernicke's area to motor planning in Broca's area.
Let's start with the one most crucial for language and spatial awareness, the superior longitudinal fasciculus, SLF.
The SLF is vast and structurally complex. It's traditionally divided into 3 distinct branches, SLF, MASKey, and 3. It runs longitudinally, connecting the frontal lobe to the parietal and occipital lobes.
And what are its main functions?
SLF 2 and 3 are critically involved in integrating visuospatial processing, working memory, and planning. Most famously, SLF-thurvin is essential for the dorsal language stream. It connects the inferior frontal gyrus, Broca's territory, to the inferior parietal lobule and superior temporal gyrus, which is Wernicke's territory.
What about the tract connecting vision and the frontal executive center?
That is the inferior fronto-occipital fasciculus, IFOF. It is a deep, sweeping bundle that connects the visual areas of the occipital lobe all the way forward to the frontal and prefrontal cortices. It's instrumental in high-speed visual object recognition and the complex, rapid integration required for reading, linking the visual word form area to executive control.
The uncinate fasciculus has a very specific shape and emotional function.
The uncinate fasciculus is named for its distinctive hook shape. It connects the temporal pole, a crucial region for emotional evaluation and memory, to the orbital prefrontal cortex for behavioral regulation.
So this tract is a vital component of the limbic frontal connection.
It is. It links primal emotional input, often involving the amygdala, with a higher-level behavioral and visceral regulation dictated by the executive brain. It allows your gut feeling to influence your moral and behavioral decisions.
Then we have the cingulum, which hugs the medial wall.
The cingulum runs deep to the entire length of the cingulate gyrus. It forms an internal ring connecting the cingulate cortex to the parahippocampal gyrus, the subiculum, and other structures of the limbic system. It is vital for integrating memory, emotional control, and autonomic function throughout the entire limbic ring.
And finally, the most historically famous language connection, the arcuate fasciculus.
The arcuate fasciculus is the large bundle of fibers that arches around the lateral fissure, connecting Broca's area in the frontal lobe to Wernicke's area in the temporal and parietal lobes. While modern research shows the SLF-third is often the primary conduit for repetition, the arcuate fasciculus remains historically crucial.
And damage to this specific tract results in conduction aphasia.
Exactly. The patient can understand language because Wernicke is intact and can speak fluently because Broca is intact, but they are severely compromised in their ability to repeat spoken words verbatim. It illustrates a breakdown of that short-term auditory motor loop.
So what does this all mean when we put the whole picture together? We have just completed a deep dive into the brain's complete architectural plan.
We really have.
We've moved from the deeply folded surface down into the six-layered architecture that defines cortical specialization, moved through the specific Brodmann areas that map function, and finally navigated the complex, dedicated fiber highways that ensure global communication.
The overwhelming takeaway for me is that the brain is not a monolithic structure. It's a meticulously constructed hierarchy of specialized processors built on a universal operating system, that six-layer rule. We saw how the thickness of those layers dictates function. Thick layer V for sensory input, thick layer V for motor output.
And we saw the evidence of evolution laid bare in the structure.
We did. The transition zones, the periarchocortex, they show the bridge between the ancient three-layered memory structures, the allocortex, and the six-layered modern cognitive structures, the isocortex.
This journey has shown us that the structures responsible for our most primal functions, fear detection in the amygdala, memory formation in the hippocampus, body awareness in the insula, they are isolated islands.
No, not at all.
They are intimately woven into our highest level processing via specific named pathways. The uncinate fasciculus, for example, isn't just a physical connector. It is the dedicated emotional pipeline linking our instantaneous emotional evaluation directly to the frontal lobe for complex decision-making and behavioral filtering.
And this brings us to a final provocative thought about what defines the human mind. We know the basic six-layer structure, the isocortex, and the columnar unit of processing are fundamentally conserved across most advanced mammals. But the truly unique aspect of the human brain is the massive size, complexity, and sheer connection density of our association areas.
Particularly the prefrontal cortex.
Particularly the prefrontal cortex and the specialized association fiber tracts like the SLF and the IFOF. So if the fundamental processing unit is the same, how does this difference in scale, this vastly enhanced ability to network disparate pieces of information, translate into uniquely human cognitive abilities?
It's a huge question.
It is. We are talking about the emergence of abstract planning, sophisticated moral reasoning, and the ability to imagine the future or understand another person's mind, theory of mind. The physical architectural difference is a difference in scale and connectivity, but the functional result is a qualitative leap into consciousness as we understand it. That scale shift remains the greatest architectural mystery we have yet to fully unravel.